Okay, so what we're talking about today is biological effects, and we're going to talk first generically about the difference between somatic and genetic effects, and then we're going to look specifically at all the different types of damage to the DNA and to the chromosomes. On Canvas for this unit, make sure that you look at the unit objectives because they outline all of the information that you need to know for this unit. And again, make sure you complete the reading, which is Carroll chapter 41, pages 761 to 765, and Statkowitz chapter 8, pages 124 to 130 and 153 to 155. So make sure that you do the reading from the textbook. All right, so when we think about radiation exposure, we know that when we expose a patient to radiation, the radiation interacts with the atoms and molecules in the patient's body and exchanges energy. That energy can be exchanged either via ionization or excitation. It can be exchanged either directly or indirectly. Ultimately, the interactions that we look at in the diagnostic energy range are predominantly photoelectric absorption and Compton scattering. So once the energy is exchanged, if the molecules and atoms are damaged, they can result in biological effects. And biological effects can be categorized many different ways, but the first way that we're going to look at to categorize the effects is to categorize them according to, are they somatic or are they genetic? So if we think about somatic effects occur in the individual who has been exposed. So for example, if you had a patient who had a very long and complicated interventional radiology case, and that interventional radiology case required 1 hour of fluoro time, that patient who receives that 1 hour of fluoro time may have skin reddening or erythema. That is a somatic effect. The individual was exposed to the radiation and the effect was seen in that individual. Somatic effects typically follow what we call a nonlinear threshold dose-response relationship. And if we look at dose-response relationships, here we see a graph, and on the dose-response relationship, the x-axis is dose, increasing as we go further to the right, and the y-axis is biological response, or the effect. Now, somatic effects typically follow a nonlinear, so it's not a line, threshold dose response. So if we look at these three examples of curves, well, 1 is a line, it's linear. 2 is a line, it's linear. 3, this sort of S-shaped, is a nonlinear curve or a sigmoidal dose response curve. All that means it's not a line. The second term is threshold. If a response has a threshold, that means there is a dose at which the response begins to be seen. So for example, we were using the example of erythema. Patient's skin's turning red after a long fluoroscopy procedure. Erythema has a threshold of approximately 2.5 gray. So below that, below an exposure of 2.5 gray, you do not see the effect. This patient's skin doesn't turn red. But once you hit the threshold, and if we say this was the threshold for skin erythema, once you reach the threshold, as the dose goes up, the response gets worse. So the dose is increasing, the response is increasing until it flattens out here at the top, meaning it can't get any worse. That's as bad as it's going to get. And we'll talk about what that means specifically in just a second. But somatic effects, effects that we see in the individual that's exposed, typically follow that nonlinear threshold dose response relationship, meaning we won't see the effect until we reach a certain threshold or a certain dose level. After we pass that threshold, as the dose increases, the response gets worse until it can't get any. And basically, it can't get any worse when we've killed all the cells. There is one big exception to this dose response curve for somatic effects, and that is cancer, okay? But all of the other somatic effects typically follow the nonlinear threshold dose response relationship. So when we look at somatic effects, we can divide them into two types. They can either be stochastic or non-stochastic. And you can find information about, or more detailed information about stochastic and non-stochastic, and about the dose response relationships in chapter 9 in the Statkowitz-Shearer textbook. I was looking for the specific page numbers. I think it is, it's like on page 160, begins on page 160. And it's at the very first of chapter 9. So if we have stochastic effects, when we think about them relative to somatic effects, the main stochastic effect that we're looking at is cancer. Stochastic effects are based on mutations. These effects are non-threshold, meaning even an individual single photon could potentially cause the effect. They are also based on probability. A single photon can cause the effect, but the greater the number of photons you're exposed to, the greater the probability of the effect. So for example, if I were exposed to a single photon to my liver, I could potentially develop liver cancer. It's not very probable or not very likely because I only was exposed to one photon. Say another individual was exposed to 1 million photons to their liver. They're more likely or more probable to develop liver cancer than somebody who only received one photon. If you had another individual that received 1 billion photons to their liver, they would be the most likely to develop liver cancer because they had the greatest exposure. So when we consider stochastic effects, cancer specifically, the cause of the cancer would be a mutation in the body cells, and the likelihood of the cancer or of the development of cancer would increase with the amount of exposure, but the severity of the cancer would not be impacted. The liver cancer that I developed from an exposure of 1 photon would basically be identical to the liver cancer that somebody developed if they were exposed to 1 billion photons. It's just that the person exposed to the greater number of photons or the higher radiation exposure would be more likely to develop it than someone who's exposed to a very low level of radiation. So again, when we think of somatic effects, the main stochastic effect that we look at is cancer induction. Radiation is a carcinogen. We'll spend the last unit in the course talking about that. And no, again, higher the amount of exposure, greater the probability of developing the condition, but no change in the severity of the condition. Now we look at the non-stochastic effects. And the non-stochastic effects are also called the deterministic effects. And they're actually now changing what they call these and going away from both non-stochastic and deterministic effects and simply calling them tissue effects. Now, I'm not sure what the rationale is for that, but that does seem to be the trend. But anyway, in all of your course documents and all of the registry materials, they would be referred to as non-stochastic or deterministic effects. And these are based on cell killing. So for example, we mentioned erythema, or skin reddening. Skin reddening occurs once you get past the threshold. So these non-stochastic effects have a threshold. As you pass the threshold, the damage gets worse and worse and worse until basically you've killed all the cells and it can't get any worse. So if we go back to this slide and we look at curve 3, once we get to the threshold, we start to see the skin reddening. As the dose increases, the skin reddening increases until it passes into a worse effect, maybe ulceration of the skin, or maybe desquamation of the skin where the skin starts to peel off. And when you get to this very high levels of radiation, you see the curve is flattened out. At that point, basically you've killed all the skin cells. It can't get any worse. There's nothing else that can happen. But with the non-stochastic or deterministic effects, they are based on that cell killing. When you've killed all the cells, it can't get any worse than that. So some examples of these non-stochastic effects, we've already said skin burns, which are erythema. Epilation, what is epilation?
Peeling? Loss of hair.
Hair loss. Hair loss. I always remember that because they sold Epilady shavers, which were supposed to remove hair. So epilation is hair loss. Cataract formation is another. So these are just some examples of non-stochastic or deterministic effects. Again, they are based on cell killing. What we're going to focus on today are genetic effects. And genetic effects are effects that are seen in unborn generations. So for example, if you have a patient who has a high level exposure, say a patient who has a high level exposure to the pelvis. Maybe it's a long-term fluoro study, whatever. The ovaries are in the field of view. Mutations could occur in the ovaries. If the eggs that are irradiated are then used in conception, then those mutations could be transmitted to the offspring. So genetic effects are effects that we would see in future generations. Because they are based on mutations, they are a stochastic effect. And they follow a linear, non-threshold dose response curve. Linear, it graphs as a straight line. So both 1 and 2 are both linear responses. Remember, 3's the sigmoidal or the nonlinear response. Non-threshold means any radiation exposure, no matter how small, can cause the effect. So it starts at 0 on the dose axis. So here we have a linear non-threshold dose response curve. Genetic effects and cancer follow this dose response relationship. They are the stochastic effects. The nonlinear threshold, number 3, that's the non-stochastic deterministic effects based on cell killing, not mutations. This second curve, which is a linear threshold, it's just thrown in there for fun to differentiate between the two. But typically, like I said, for the stochastic effects, cancer and the genetic effects, linear non-threshold. For the non-stochastic effects, non-linear threshold dose response. Okay, so does anybody have any questions related to the difference between somatic and genetic effects and the difference between the different dose response curves?
I do. So in my notes under somatic effects, I have stochastic and non-stochastic effects. And
Yes.
then under genetic, I have just stochastic. That's correct?
Yes, that's correct. And because somatic simply means of the cells of the body. The individual's exposed, they're going to see the effects. So if I'm exposed to high-level fluoro, I may develop erythema. Erythema is a non-stochastic effect. Based on cell killing, I have to reach a threshold dose before I'm going to see my skin turn red. Okay? Cancer is the main stochastic effect that is somatic. If I could be exposed to a single photon theoretically and develop cancer. It's not likely, but it could happen. So it is a stochastic effect, but is also a somatic effect. Does that make sense?
Yep.
Okay. Anybody else have any questions? No questions?
Yeah.
All right. So we're going to continue on.
Oh, wait, hold on one second. You said genetic effects is stochastic effect only, right?
Yes, genetic effects are considered stochastic. And they're, an individual is exposed, their reproductive cells have damage via mutations. And then if they are used in conception, that's when the genetic effects would be expressed.
Okay.
Okay?
Yes, ma'am.
All right. So we're going to start now, which really is the focus of this first segment, and it's to look really at the genetic effects more closely. So when we think about genetic effects, we should automatically think of the DNA, genes, and the chromosomes, because those are the things that control our heredity. So we're going to start just briefly reviewing the DNA structure, and then we're going to look at, like I said, the function of the DNA, and then the function of the genes and the function of the chromosomes, and what happens when they are irradiated. So if we irradiate our cells, we know the DNA is the controlling molecule in the nucleus of the cell. It controls metabolism. It controls all of our genetic information. So genetic effects are due to structural damage to the DNA molecule. Now, to review, it's important that you understand the DNA structure. So we know that the DNA is this double helix configuration. It's like a twisted ladder. The side chains, or the side rails, are sugar phosphate chains, alternating sugar and phosphate molecules. The nitrogenous bases branch off of the sugar molecule. How many nitrogenous bases are there?
Four.
Four, okay. What are they?
Adenine. Oh, it's right there. Adenine, thymine, guanine, and cytosine.
All right, it's right on the slide. Adenine, thymine, guanine, cytosine. Those are your four nitrogenous bases. The base pairs can only join in specific ways. So what two pairing combinations are possible?
TA.
A and T.
Adenine and thymine.
Yeah, A and T.
A and T, adenine and thymine, and that leaves guanine and cytosine. Okay, so those are the only allowable base pairs. So if we look at the DNA molecule, again, the side rails are the sugar phosphate chains. The nitrogenous bases branch off of the sugar, and they can pair A and T or G and C, and the base pairs are held together by a hydrogen bond. If we look at one small unit of the DNA molecule, it's called a nucleotide. So a deoxyribonucleotide. As we've just said, y'all remembered from previous lectures, the allowable base pairs, A and T, G and C. And here's a new term. The term is a codon. A codon is one bit of information on the DNA molecule. You can think of a codon, for example, as analogous to a word in a sentence. So a codon is a sequence of 3 nitrogenous bases. So if a scientist wanted to decode the DNA molecule, they would literally start at one end of the molecule and look at the sequence of the first 3 bases. And that's one bit of information. That's a codon. Then the next 3 bases, that's another piece of information, another codon. And they would have to look at each sequence of 3 nitrogenous bases to decode the molecule, to determine all the different things that the DNA molecule does. Any change in a codon changes the performance of the molecule, or changes the outcome of what that molecule is supposed to be doing. So that's an important point to remember. So what is the function, or what are the functions of the DNA? Well, one, we've said the DNA carries all of our genetic information. The second thing is that the DNA contains information for replicating itself. Remember when we talked about the cell cycle and we had M phase where the cell's dividing, and then G1 for RNA synthesis, then S phase for DNA replication. The DNA has to replicate in order for cells to divide. Number 3, the DNA mutates and then it replicates the mutation faithfully. A mutation simply means any change in the base sequence on the DNA molecule. When that happens, again, it's going to change the outcome of the molecule. And mutations do happen all the time, spontaneously. So when a mutation occurs, whether it's induced or it's spontaneous, it must be replicated faithfully. And the last is that the DNA must have a way to decode that stored information in the molecule and translate it into action. It must be able to transmit information, say, to the messenger RNA, so that the messenger RNA can go into the cytoplasm and cause protein synthesis to occur. So all of these are the functions of the DNA. It has to be able to do all of these things. Now we've already said this, that the sequence of bases on the molecule are critical. And those sequence of bases, every 3 bases is called a codon. And that is what determines your hereditary information, and it determines your metabolism. If a codon is changed, if a base is lost, if a base molecule is changed, that's referred to as a frame shift mutation. Frame shift mutations are typically lethal to the cell, so that makes it a big deal. So anytime the base molecules, remember, a sequence of 3 bases is making up a codon. If any one of these base molecules is either lost or changed, that changes the codon and changes the expression of that portion of the DNA. And the term, and this term is in the Carroll text, it's a relatively new term. It's called, again, a frame shift mutation. All right, now a little bit later we will look at all the different changes that radiation can cause in a DNA molecule. But now we're going to shift to the genes. So the DNA makes up the genes make up the chromosomes. The genes are considered the basic unit of heredity. So this is what's transmitting all those different traits that you receive from your parents, your genes. So here's a question. How many genes do we have?
23. 23.
You're thinking chromosomes. The genes are on the chromosomes, but how many?
46.
Nope, you're thinking chromosomes too.
Thinking thousands.
Thousands.
Millions.
Anybody else want to guess? Have any of you ever heard of the Human Genome Project?
Yes.
Okay, so the Human Genome Project has been around for many years now, and they were tasked with decoding the DNA and determining how many genes that we have and what genes control what. Well, here's what they found. Going in, they thought that a human genome was about 100,000 genes. They decided that no, it was much less than that, maybe only 25 to 30,000 genes. They found out that a lot of the DNA are really not genes, but they are switches that control turning on and off particular genes. So the answer is they're not exactly sure how many genes we have, but probably somewhere between 25 and 30,000. The sad truth is we don't really have that many more genes than an earthworm, which I thought that was interesting, you know, since we're a higher being, I guess. But there's still a whole lot of the DNA that they don't know what it does. Some of it, like I said, I mentioned some of the DNA deals with switching on and off certain genes. They call that the dark matter. I'm like, okay, that sounds like something from science fiction. But the dark matter, they don't really know a whole lot about it. So it's still an area under investigation. They're not exactly sure what all of the DNA is doing, even after years of study. But anyway, we're going to look at genes from a very general standpoint to start with. And again, we'll come back later and talk about radiation-induced damage to the genes. So the genes are our basic unit of hereditary information. We know they're composed of DNA, and the genes make up the chromosomes. If we look at a chromosome, here's an illustration of a chromosome. This central portion of the chromosome is the centromere. And then we have supercoiled DNA in the chromosomal proteins, and that's what makes up the entire chromosome. Well, certain segments of DNA that occur in very specific locations on the chromosomes are the genes. And your genes occur in complementary pairs. You get one gene from your father, you get one gene from your mother. So for example, for hair color, you may have a gene from your mother for blonde hair and a gene from your father for dark hair. You know, what color is your hair? Well, that would depend on which is dominant. We'll talk about that. But for
square
traits, you get one gene from each parent. And when we talk about our genes, we can talk about them in terms of phenotype or genotype. Your phenotype is the observable characteristics that you have, and your phenotype is based on your genotype. Your genotype is your complete genetic makeup. But your phenotype is how you look and how you act, even mannerisms. So do any of you have siblings? Any of you have brothers and sisters? Okay, several of you have brothers and sisters. Think about a trait that you have, a phenotypic trait that is similar to your sibling or a sibling, and then relate it back to one of your parents. Because remember, you're getting your genes from your parents. So for example, I'll give you an example. I have 4 sisters. We all sound very much alike. I don't think we look that much alike, but we sound alike. And we sound like our mother. So our voice, the tone of our voice, our expressions sound like our mother. All right, any of you want to share?
I look like my mom.
Okay, you look like your mom? So your facial features, your facial structures?
Yep.
Do you have siblings? Do they look like your mom? They look like you?
Only child.
Your only child, okay. All right. Anybody who have siblings, do you have any characteristics that you share with your siblings?
We're all slim.
Me and my brother have the same face.
Courtney and her brother have the same face. Is that what you said?
Mm-hmm.
Kiara says her family's all slim. That's a good trait to have.
Me and my brother are like polar opposites and look nothing alike. He's 6'1". I'm 5'1". He's like, we don't look anything alike.
Do you look more like your mom or your dad, and does he look more like one or the other?
He doesn't look like either parents. He looks like my mom's brother, but
Okay.
I look like my dad.
Okay, interesting.
He has like bright blue eyes, and I have brown eyes. It's weird.
Mm-hmm. My daughter looks more like my husband. My son doesn't look like either of us, but he looks like one of my uncles, which is I find strange. But he doesn't look like really me at all. But obviously he has some of the traits of my mother's family. So genetics are interesting. Genetics are an interesting thing. I always wonder, what causes you to change? My eyes were blue until I was about 12 or 13, and now they're a greenish gray. And y'all won't believe this, when I was born, when I was young, my hair was white blonde and curly.
Really?
And now it's this. What happened? Where did my blonde hair go? Where did my curly hair go?
Did you dye it or do something?
No, I got old. Who knows what happens?
I thought it goes gray. I thought that means it's supposed to go gray.
Oh, it is going gray, but you just can't see it very much. But
I was just going to say that happened with my nephew. He had he had blondish curls when he was a baby, and they were all blonde, straight curls. So cute. And then when he got older, it's like the texture of his hair and the color changed, and it's more sandy brown right now. Mm-hmm. He's in his 30s, but it was completely blonde when he was a baby.
Yeah. My hair has gotten darker literally every year of my life. I said, I'm going to be the only 80-year-old woman with coal black hair because it gets dark all the time. But I think
my eyes changed color.
Marquise, what color were they?
They were like gray blue.
And
now I have no idea. Sometimes they're brownish looking, sometimes they're greenish looking.
Yeah. I just think that kind of stuff is interesting, and why it happens. I mean, I don't really know.
My moms are like Markevia's. They have changed about 4 times. Now they're like a mixture of all 3 colors. It's kind of beautiful, but strange. They've changed a lot.
So anyway, all of those things we're talking about are phenotypic expressions. Our genetic makeup is all of the different genes that we have. And the genes that we have control our phenotypic expression. If you have genes that are the same, for example, the example here is with hair. If you received 2 genes for red hair from each parent, or one from each parent, the genetic material is the same, and that's termed homozygous. So you have homozygous genes for hair color if both of your genes are the same. So if you had 2 genes for red hair, you have a complementary pair of homozygous genes. If you had 2 genes for blue eyes, then your eyes are going to be blue, and those genes again are homozygous. They're the same. If, however, you get 2 different genes for the same trait, maybe you have a gene for red hair and a gene for blonde hair, those are heterozygous. The genetic material for each complementary gene is different. If you had 1 gene for blue eyes and 1 gene for brown eyes, you would have heterozygous genes for eye color. So the genetic material on the complementary pair is different. So that being said, you know, if the genes are the same, it makes sense that you're going to have either red hair or blue eyes or whatever. If the genetic material is the same, that's going to be the trait that's seen. But if they're different, if you have one gene for blue eyes and one gene for brown eyes, what color are your eyes going to be? Well, the answer is it depends. It depends on which genes are dominant and which genes are recessive. Dominant genes are genes that are always expressed, whether they're in a homozygous state, meaning both genes are the same, or in a heterozygous state where they're different. So for example, using the eye color, brown eyes are dominant. That's a dominant trait. So if you received a gene for brown eyes from your mom and a gene for blue eyes from your dad, you're going to have brown eyes because that is a dominant gene. If blue eyes is a recessive trait, you can only have blue eyes if it's paired with another gene for blue eyes. So recessive genes are only expressed if they're in a homozygous state. If you got a gene for blue eyes from your mom, a gene for blue eyes from your dad, they're in a homozygous state, you're going to have blue eyes. If blue, again, is recessive, if you got a dominant gene for any other eye color paired with that recessive gene for blue eyes, then you're going to have some other eye color. So is everybody okay with dominant and recessive genes, homozygous, heterozygous states? Are we all good with that?
Yes.
Okay, great. So now we go to the chromosomes. Now the chromosomes, again, the DNA makes up the genes that makes up the chromosomes. The chromosomes consist of supercoiled DNA in chromosomal proteins. So the chromosome is simply one huge strand of coiled up DNA from top to bottom that's held in place by those chromosomal proteins. Remember, the attachment in the center is the centromere. The things that extend from the centromere are called the arms of the chromosome. This is a chromosome that's been duplicated. So these entire structures are the chromatids, okay? So centromere holds it together. All of these are the arms of the chromosome. And again, a single one would be a chromatid. Uh-oh, sorry, I need to go back. We have 23 pairs of chromosomes. We have 22 somatic chromosomes that are called the autosomes. And then we have one pair of genetic chromosomes. So here we see a map of the chromosomes. At what stage of cell division are the chromosomes most visible? When do they line up across the equator of the cell?
Anaphase?
Metaphase. Metaphase. So here we have the chromosomes. Here we have them paired and lined up. Uh-oh, sorry. Does anybody know what we call that when we've mapped out the chromosomes? Anybody know? It's called a karyotype. K-A-R-O-T-Y-P-E, a karyotype. These first 23 pair, we see, first 22 pair, excuse me, are the autosomes. The last pair are the sex chromosomes. So looking at these chromosomes, are these the chromosomes of a male or a female?
Male.
Male. How did you know that?
There's an X and a Y.
There's an X and a Y. If it were a female, there would have been 2 X chromosomes, but it's an X and a Y. So this is a male. So this is a karyotype of a male's chromosomes. And again, the first 22 pair are the autosomes. The 23rd pair are the sex chromosomes. So we have 23 pairs or 46 total chromosomes in a set. So what are the function of the chromosomes? Well, the chromosomes only have 2 jobs. One is to maintain the proper order of the genes. So remember, your genes are at specific locations along the chromosomes. The chromosome's job is to keep them in that proper order. And the second job of the chromosomes is to maintain the correct amount of genetic material during cell division. Remember the cell cycle. In S phase, the DNA replicates, so there's twice the amount of genetic material. In cell division, the chromosomes split and half goes to each new daughter cell. So the function of the chromosome is to make sure that the correct amount of genetic material gets to each daughter cell during cell division. So there's some terms we want to mention. First, we're going to mention the diploid number and the haploid number. And I think we've already mentioned this, but the diploid number basically means that cell has 2 sets of chromosomes, or they have 23 pairs. That is normal for all somatic cells. All somatic cells should have 23 pairs of chromosomes. The haploid number is basically half the diploid number. So instead of having 23 pairs, you only have 23 chromosomes. That is normal for genetic cells, because at conception, half of the chromosome, those 23 chromosomes from the father join with the 23 chromosomes from the mother to give you a full set of chromosomes. So when we talk about the diploid number, we're referring to the normal number of chromosomes for a somatic cell. And if we're talking about the haploid number, we're talking about the normal number of chromosomes for genetic cells. Okay. Some additional terms. Euplody. Euplody simply indicates that this person has a complete set of chromosomes. They've got the normal number of chromosomes that they should have. Aneuploidy means they don't. Aneuploidy is a general term that simply means they have an abnormal number of chromosomes. They could have too many, they could have too few, but they don't have the proper number. Polyploidy, poly meaning many, is a term that we use when the person has more than the normal number of chromosomes. So euploidy, you've got the normal number, the normal amount. Aneuploidy, you don't. It could be too many or too few. Polyploidy, you've got too many. Okay, so let's look at the karyotype on the left. Can somebody find the problem
Is it in the sex genes?
What do you think's the problem there? And it's not the genes, it's the chromosomes.
I mean the chromosome, sorry, yeah.
Too many of the
Too many,
X
and
Y.
They have too many.
Yeah.
There should only be 2 sex chromosomes, but there are 3.
So they are
2 X's and a Y.
So they're polyploidy, what do you call it?
They have polyploidy.
Polyploidy, yeah.
And euploidy. They don't have the normal number. More specifically, they have too many. So they have trisomy, which is 3 sex chromosomes. So
what does that mean for them?
This would actually be a male because it's got a Y. It would be a male with more
have a lot of
characteristics because they have 2 X chromosomes.
Oh wow. Okay, yeah. Sorry, this is interesting.
I can't remember if it's Turner syndrome or Klinefelter syndrome. It's one of the 2. I can't remember. But it would be a feminized male, basically.
Wow.
Okay, so look at the karyotype on the right. So what's the problem there?
Their chromosomes look weird. Like they're not, they look
Well, they may look weird. Look specifically for number.
Maybe because
21, they have too many.
Aha, look at 21.
Yeah.
They have 3 chromosomes at the 21st site. So this is trisomy 21. It's also aneuploidy. They've got an abnormal number. It's also polyploidy. They have too many. This is a very common condition. So trisomy 21, what is it?
Is that Down syndrome?
It is Down syndrome. This is Down syndrome. Okay, so let's pause here and let's take a break for. Okay, so we have looked at the basis of our genetic material. We've reviewed the structure of the DNA. We know that the DNA makes up the genes make up the chromosomes. And the chromosomes are to make sure our genetic material, our genes stay in the proper order and to make sure that we get the proper amount of genetic material during cell division. So the question becomes, what happens to our DNA, our genes, and our chromosomes when they are irradiated? And the answer is, well, mutations. So we're going to start by looking first at the effects of irradiation on the DNA. And when radiation interacts, we know it interacts at the atomic and molecular level, and it can cause changes in the molecule. Well, any change in the DNA molecule is a mutation. Point mutations are described as single chemical changes. It's been refined a little bit now. They're looked at as changes in the bases, not the side rails, but in the bases of the DNA molecule. We should be specifically concerned with point mutations because point mutations are thought to occur from chronic low-level exposure. Now, chronic low-level exposure to radiation is the type of exposure that we receive from occupational exposure. So as part of our job, we are working with patients, administering ionizing radiation to patients. Some of that radiation will scatter and it will expose us. So as a part of our career, we are exposed to chronic low-level radiation, and chronic low-level radiation is one source of point mutations. So a mutation simply indicates a change. But when we change the DNA, we can change the expression of the gene. Because remember, each sequence of 3 bases on the DNA molecule is a codon. And if we change that information, if we change even a single codon, then we can change the expression of the gene. And remember, anything that changes the base molecules or the rung of the DNA ladder is called a frame shift mutation. And when we have a frame shift mutation, that can change the genetic code. So that's the bad news. The radiation can cause mutations that can change our genetic code. The good news is that our bodies have a remarkable ability to repair. And that is what really is so phenomenal. 90% of radiation-induced damage is repaired. So even if we are receiving scattered radiation as part of our occupational exposure, 90% of the time the body is repairing it. So that's the good news. So what are some of the types of radiation damage to the DNA? Well, there's base damage, single-strand breaks, double-strand breaks, cross-linking, and rung breakage. So we're going to look at each of these. Remember, when we talk about the strands, we're talking about the sugar phosphate side rails. When we talk about the rungs or the bases, we're talking about the 2 nitrogenous bases held together with a hydrogen bond. So we'll start with base damage. If one of the nitrogenous bases is irradiated, it can either change the molecular composition of the base, or it could potentially delete the base from the molecule. It could break it off from the side chain. This is a mutation. It is a point mutation, and it will change the codon. Remember, the other name for it also is a frame shift mutation. So a point mutation, a frame shift mutation, it will change the codon, and therefore it will change the expression of that gene in that area. Typically, base damage is lethal to a cell. The cell may be able to divide, but the daughter cells would more than likely have a bad effect from that loss of base. So base damage is a big deal. Then we have single-strand breaks. Single-strand breaks are also called main chain scission. That just means we have broken the sugar phosphate chain. So if you look in the illustration, here we see a break in the sugar phosphate side rail. Main chain scission is literally of little consequence. The body can repair. It's usually an easy repair, and usually it's no big deal in terms of the survival of the cell type. So if we have a single strand break, not really a big deal. It can be easily repaired. Then we have double strand breaks. And double strand breaks, obviously, is when we have a break on both side rails. So here we have, here we have a break on this side, and then down here we have a break on the other side. Double strand breaks are of little consequence as long as they are separated on the side rails. So for example, this one is way up here, this one's way down here. They're separated in distance. These would be treated like two single strand breaks. They would be repaired, not a big deal. Double strand breaks become a big deal when they occur at the same level. So for example, if there was a very highly ionizing radiation, such as alpha particles. Alpha particles, remember, are high LET radiations. They are imparting a large amount of energy along a very short particle track. So alpha particles could break both side rails of a DNA molecule because they have so much energy. Now, if the molecule does not split, again, they will be repaired. But the problem here is that the molecule can be broken literally into two pieces. So if the molecule is broken into two pieces and separates, that damage can also be lethal, because essentially you've lost half the DNA molecule. So there is an increased correlation with cell killing if the double strand break is at the same level and the molecule splits into two pieces. If they are at separate levels, such as shown here, they're usually treated as single strand breaks and are of little consequence. The next thing we have, or the next type of damage, is called cross-linking. Now, those chromosomal proteins are sticky. So when things break apart, they can rejoin with other segments where they shouldn't rejoin. And that's when cross-linking occurs. Cross-linking occurs when a broken portion of the DNA molecule, specifically the bases, attach to the same molecule in an incorrect place, or they attach to an adjacent DNA molecule. So if, as in this illustration, so there's a break and then this base joins with this base down here. That is cross-linking, and it is called an intra-strand cross-link because it's joining in the wrong place to the same DNA molecule. So it is an intra-strand cross-link. If the bases were broken apart and this base joined with an adjacent DNA molecule, it would be called an interstrand cross-link because it's between two adjacent DNA molecules. Obviously, the codons have been changed. Things aren't joined where they should be. So this also could be lethal to the cell. And then we have rung breakage. Remember, the rungs of the DNA are the two bases joined together by the hydrogen bond. Rung breakage occurs when the hydrogen bond is broken between the two bases. This type of damage is relatively easy to repair, so it is also not of any major consequence. So any of these changes are considered mutations. Mutations are typically harmful, and they're also typically recessive. So if a mutation occurs, it may not be expressed phenotypically because it's typically recessive. However, again, they are typically harmful, meaning something's not going to function as normal. The one thing to note is that radiation-induced mutations are no different from mutations that occur spontaneously. Radiation is not going to produce any freaks of nature. It's not going to produce a Ninja Turtle. It's not going to produce Godzilla. It's not going to produce the Fantastic Four. Radiation does cause mutations, but they are the same types of mutations that would either occur spontaneously or from other causes that we'll look at. And when we think about radiation-induced damage to the DNA, the most severe damage occurs if you have one of those frame shift mutations where the base molecule is either damaged or deleted. Okay, so we know radiation can cause mutations. It can cause, or it can cause a change in the expression of the gene, which is phenotype, but it may not. But it will always change genotype. So here we have a mutation. Look at this image. Anybody know what that's called or what the problem is? How would you describe this problem?
I thought it was just fused fingers.
No, well, give me a medical term.
I used to know.
You used to know.
Yeah.
Well, what's the first thing you know? Look at the number of fingernails.
They got 3.
Is it
Six. Okay.
Polydactyly?
They have 6 fingers. What'd you say, Markavia?
Is it like poly something?
I was saying
Polydactyl?
polydactyly is what I said. Is it
Okay,
that?
polydactyly means more than the normal amount. And so, yes, we're seeing 6 fingernails. I'm not really sure if they're 6 finger bones because we have no x-ray. The fact that they're fused is called syndactyly. So polydactyly, we think they may have 1 extra finger. Syndactyly, at least 3 of those fingers are fused. So this is a mutation. Did it occur spontaneously? Was it induced by radiation? Was it induced by something else? We don't know. This is just a random picture that I found. But mutations do occur spontaneously. That just means they occur for no apparent reason at random. They're occurring all the time, but it's a relatively low rate of occurrence. We also know that we can induce mutations. That means we can cause them with specific things. Radiation, chemicals, and viruses are known mutagens. So if you exposed a large population, say such as Hiroshima or Nagasaki, when they were exposed, a large population, to radiation, we would expect an increased rate of mutations occurring. And again, just what I said previously, induced mutations are not going to show up as something very different or something we've never seen before. It simply increases the rate at which spontaneous mutations would occur. Down syndrome is a result of a mutation. Irradiating a large population might cause an increase in Down syndrome children. So that would be an example.
I have a question. I can
Yes.
kind of remember from one of Markevia's presentations about a mass-like thing in radiation, hydrocephaly, but it was a while ago. Yeah. Am I correct? Is that part of mutations that can happen from radiation?
Pretty much anything can happen. If that's something that would have happened, can happen spontaneously, yes, it can be radiation induced. One of the things, to give you an example, I saw a documentary several years ago about a small town in, I want to say it was Pennsylvania, that had a Polaroid plant. And the Polaroid plant made Polaroid sunglasses. And over the years, chemicals from the plant had been leaking into the water supply. And there was a college, not a college, it was a high school class reunion. All these people had graduated from high school, had moved away, married, had families, and they had come back for this class reunion. And what they found is that in their class, relative to the normal population, a huge percentage of them had autistic children. Some of them more than one autistic child. And they started to investigate. And what they found was that the contamination, the chemical contamination from that Polaroid plant, it had contaminated the water supply. And so they had been exposed. When they lived there from the water supply, it had damaged their reproductive cells, and their children were autistic. A lot of their children were autistic. So that's an example of a chemical mutagen. So autistic children occur all the time for no apparent reason, but that wasn't a case where they attributed it back to the chemical contamination of their water supply. Radiation does the same thing. Radiation can cause mutations that would cause anything that normally occurs. Like I said, it's just not going to produce Godzilla or Ninja Turtles.
Okay, so when somebody is in an area where they have like high altitude, they develop over time the ability to like take that altitude better than other people, and it's a change. Is that a mutation?
That's an environmental compensation. It's compensatory polycythemia compensatory polycythemia. They produce more red blood cells to deal with the lower oxygen levels. So no, that's not really, that's more an accommodation or an adaptation from it. All right, so we know what radiation can do to the DNA. We know that any change to the DNA results in mutations on the chromosomes. And we know that, or mutations on the genes, excuse me, and we know the genes make up the chromosomes. So what can happen to the chromosomes? Well, radiation can damage the chromosomes, and this damage is called different things depending on which textbook you might want to look at. Sometimes it's referred to as chromosome aberrations. Sometimes it's referred to as chromosomal lesions. Sometimes it's referred to as chromosomal anomalies. They're all the same thing. They simply mean that the chromosome has been damaged by radiation. Now, the chromosomes are damaged in one of two ways. Number one, pieces of the chromosomes can break off, and two, the genes can be rearranged. Now we know that, remember, one of the main jobs of the chromosome is to keep the genes in the proper order. So gene rearrangement or breaking off and losing part of the genetic material on the chromosome is a big deal, because cells that do not have the proper number of chromosomes nor the proper order of chromosomes are typically not viable, which means they are going to die either before they try to divide or during cell division. But again, there is some good news. Remember, the body has many, many repair mechanisms. So if we have radiation that breaks the chromosome, so here we have an illustration, here's a chromosome, there's the centromere, here are the arms. Radiation breaks off a chunk of the chromosome. 90% of the time, restitution occurs. And restitution simply is the repair of the chromosome so that the chromosome goes back to normal. So yes, radiation can damage the chromosomes, but 90% of the time, that chromosomal damage can be repaired. So let's look at what happens in those other 10% of the cases. Okay, so we said that damage to the chromosome can consist of breaks, and if the chromosome breaks and does not repair, that means we are losing genetic information. So here we have an illustration where we have two chromosomes that are irradiated and both ends of the chromosomes are broken off. So here we have most of the chromosome with a centromere, and then we have these pieces. These pieces are referred to as acentric fragments. An acentric fragment is a piece of chromosome without a centromere. It will be lost in cell division. Remember, in cell division, where the spindle fibers connect to the centromere and pull it across the cell so that we end up with the appropriate amount of genetic material to each of the daughter cells. Well, in these cases where we're missing a large chunk or an acentric fragment, they don't have anything to attach to the spindle fiber. So they're going to be lost. So that means the cell may not function properly because it's lost part of its genetic material. And when it divides, it will die because it will not transfer the proper amount of genetic material during cell division. Excuse me. Okay. The other thing that we said could happen to the chromosomes is that the genes could be rearranged. And remember, I told you earlier, the chromosomal proteins are sticky. So they will stick to each other, they'll stick together. So here's some things that can happen. The first is translocation. So we have two chromosomes that are adjacent to each other. Irradiation breaks off the ends of each chromosome, and then the body tries to repair. But in its repair, it joins each end to the wrong chromosome. So that's called a translocation. They may or may not have all the genetic material, but it's obviously not in the right order because the genetic material from one chromosome is now on another. Another type of gene rearrangement is called inversion. Inversion occurs when a chromosome is broken at two places, and in the repair, the gene somehow flipped around. You see a chromosome, a normal chromosome, and it's illustrating the order of the genes as A, B, C, D, E. Well, we irradiate the chromosome and it gets broken in two places. This center part, B, C, and D, somehow get flipped over and the chromosome repairs, but the genes are now in the wrong order. So instead of saying A, B, C, D, E, now it's A, D, C, B, E. So we have the correct amount of genetic material, but it's not in the right order. And cells that have genetic material in the improper order are also typically not viable. Another form of gene rearrangement is simply called a ring chromosome. So if a chromosome is irradiated and both ends are broken off, we now have the central part, and then we have these two acentric fragments. Well, sometimes in the efforts to repair, the two ends of the longer part rejoin, forming a ring, and then the two acentric fragments will rejoin. So we have a large portion of the chromosome that has no centromere, so it's going to be lost in cell division. And then we have the majority of the genetic information in some configuration that the cell doesn't understand. So again, when the genes rearrange, cells are not viable. The next type of gene rearrangement is called a dicentric chromosome. And a dicentric chromosome occurs when the broken ends of two chromosomes join together, giving you one chromosome location with a lot of extra material and two centromeres, and then other acentric fragments just floating around. So for example, you had the ends broken off of two chromosomes, you had two acentric fragments that are lost, but then these two larger chromosomes join together. Here's an example where we see two centromeres. So that is a dicentric chromosome. You've got excess genetic material and two centromeres at one location, and at another location, you basically have just acentric fragments floating around. The last of the radiation-induced effects on the chromosome is simply called clumping. After high levels of irradiation, sometimes the chromosome simply collapses onto itself into like just a sticky ball. And again, your body doesn't really know how to deal with that, so the cell is not viable. All right, so things to remember. Number one, 90% of damage to the chromosomes can be repaired, so that's good. But if the damage cannot be repaired and we have lost genetic information, or if the genes are out of order, then the cells will typically will typically die when they try to divide or during cell division. So if we think about the effects of radiation on the chromosome, it's important to think about what influence does dose, LET, and dose rate have on chromosomal damage. Well, from what you've learned earlier in the semester, it's pretty much common sense. As dose increases, and this is a measure, this is a graph from an article that was measuring the number of dicentric chromosomes, those with two chromosomes, two centromeres, excuse me, versus dose. And we've got different kinds of radiation. We've got gamma rays, we've got X-rays, we've got neutrons. So we've got all different energy X-rays, but we have increasing LET radiation. So if we look, the higher the dose, the greater the number of dicentric chromosomes for every type of radiation. And it's considered a linear quadratic relationship. You see how it's kind of curved. But just in general, dose goes up, damage to the chromosomes go up. If we look at LET, X and gamma are low LET radiations, neutrons are high LET radiations. So as LET goes up, the amount of chromosomal damage goes up. And it's not shown here. Oh, it is shown here, excuse me, dose rate, because these are at 0.5 gray per minute. This is 1 gray per minute. As dose rate goes up, chromosomal damage goes up. So this really just confirms what we know from previous lectures. The greatest amount of damage is done to any body structure with a higher dose from a higher LET radiation and at a higher dose rate. All right, so we've looked at the chromosomes themselves, but now we're going to take just a moment and look at the effects of radiation on the centromere. Now remember, the centromere is what is connecting the arms of the chromosomes. So when we irradiate a cell, one of the things that can happen to a chromosome is that one entire arm of the chromosome can be removed. And that would be called terminal centromere. So you have the centromere and one arm of the chromosome, and then you have basically the other entire arm of the chromosome is an acentric fragment. So obviously that is lethal to the cell. Another thing that can happen when the chromosomes and the centromere are irradiated is called chromosome lagging. And again, we have to think back to cell division to understand the importance of the centromere. Remember, one of the jobs of the chromosome is to get the appropriate amount of genetic information to each daughter cell. So if cells are irradiated, during the next cell division, the spindle fibers attach to the centromere duplicates, and then each chromosome should move along the spindle fiber to get to the appropriate location in the daughter cell. Well, sometimes after irradiation, the chromosome doesn't move at the appropriate rate along the spindle fiber. It moves slower. So if it moves too slow, it doesn't get to the opposite pole of the cell before the nuclear envelope reforms. When that happens, you have a chromosome left out in the cytoplasm. It's a loss of genetic material, and that too is lethal to the cell. The third thing that can happen to the centromere is called non-disjunction. And non-disjunction also is an issue that occurs during cell division. And let's look at the term. What is a junction? Well, a junction is a joining, okay? So if there's a junction, it's a joining, maybe like a joining of two streets. Disjunction means not joining. And so if we have a non-disjunction, that means it doesn't not join, or it doesn't break apart. So if we relate this back to the centromere, you're like, well, that was a whole convoluted way to get to this. But again, think of the job of the centromere. During cell division, when cell division starts, the chromosomes are duplicated, but there's only one centromere. So during the stages of cell division, the centromeres duplicate and the spindle fibers join to each of the separate centromeres and then pull the chromosomes to the appropriate side of the cell. In non-disjunction, the centromeres don't break apart. So the spindle fibers are trying to pull the chromosomes to the opposite poles of the cell, and they're just sort of stuck out there in the middle playing tug of war. The centromeres won't break apart. So what does this mean? Well, sometimes if they're just stuck at the middle part of the cell, neither daughter cell gets any of the genetic material. So you have two daughter cells both missing genetic material. Sometimes one spindle fiber wins out and pulls both of the chromosomes to one daughter cell. So one daughter cell has too much genetic information and the other has none. Okay? In either case, both cells have an abnormal amount of chromosomes. So here's the example where we have trisomy or monosomy. Remember we mentioned Down syndrome? Down syndrome is trisomy 21. Down syndrome is a result of non-disjunction. Down syndrome is most commonly associated with an aging mother. You know, mothers over the age of 35. But it can happen really at any age mother, but it's just one example of an event that occurs spontaneously, but can also be radiation induced. So again, if we think about the chromosomes, we're mostly concerned with losing genetic material, the rearrangement of the genes. If we think about the centromere specifically, the most common issue with the centromere is that the proper amount of genetic material doesn't make it to the daughter cells. And again, if the proper amount of genetic information doesn't make it to the daughter cells, then the cells are not viable. All right. The last section that we're going to deal with looks at how do we talk about all of these different mutations in terms of the broader population. Instead of looking at one individual, what does it mean for the entire population? And so there's some ways to express that. And the first term that we mention is the genetic load. Now, the genetic load is the total number of bad genes in the population. You know, mutated genes, problem genes, whatever we want to call them. Well, the genetic load depends on the mutation rate, how frequently mutations are occurring, and then gene removal by natural selection. Remember, mutations, radiation-induced mutations specifically, are typically recessive, not dominant. So they're typically not expressed phenotypically, but they can still remain in the gene pool. Typically, the population should be in a state of what's called genetic equilibrium. If the population is in a state of genetic equilibrium, that means the rate at which mutations are produced is equal to the rate of removal. So it's a balance. But there are things that can throw you out of balance. For example, the atomic bombs of Hiroshima and Nagasaki. That was a large-scale radiation exposure to a large population. The Chernobyl nuclear power plant accident, where that cloud of radioactive material was spreading all over Europe, exposing a large population. So those types of events would set the population out of genetic equilibrium, because radiation is a mutagen. So the mutation rate would be higher than the rate of mutant gene removal. It's important to note that since radiation-induced mutations are recessive, they would remain in the gene pool for longer periods of time because they might not be expressed. If it were a dominant mutation, it would immediately be expressed. The organism might not survive because of that mutation. But if it's recessive, it could go many generations before it was ever expressed, so it would remain in the gene pool for a long period of time. So, how do we talk about these changes in the number of mutant genes in the gene pool and how much radiation it takes to cause them? Well, the way that we talk about it are using one of the ways that we talk about it is using the concept of doubling dose. And the doubling dose is the radiation dose that would basically double the spontaneous mutation rate. So if you gave the doubling dose to an entire population, you would expect twice the number of mutations to occur. Well, how much is the doubling dose? Well, over the years, the estimates have changed. UNSEER, which is the United Nations Scientific Commission on the Effects of Atomic Radiation, estimated it in 1977 at 100 rads, which is 1 gray. BEER-5, which is the Biological Effects of Ionizing Radiation Commission, in 1980 said, well, it could be anywhere from 50 to 250 rads or 0.5 to 2.5 gray, depending on how it's administered, either acutely or chronically. Later reports in 1990 and 2006 from the BEER Commission have basically reiterated that it's still approximately 1 gray. So if a large population received an exposure of 1 gray, we would expect the mutation rate to double. There's another concept that's called the genetically significant dose. And the genetically significant dose really was trying to determine what is the effect of medical exposure on people who are childbearing age in terms of genetic impact. You know, what dose of radiation would individuals who are childbearing age have to receive in order to have an impact on the mutation rate? It is an estimation, and what they have determined is that it's estimated at 17 to 20 MR. Can anybody convert that to milligray for me? Anybody know what MR is in terms of milligray or millisievert? Anybody know the conversion factors?
Isn't there it's either 100 or 1,000 rads to a sievert?
To a gray. And it's 100. There's 100 rads to a gray. There's 100 rem to a sievert. So if this is 20 MR, it's 0.02 R, which we would consider rem. If we convert that, oh gosh, I have to do it on paper. I can't do it in my head anymore. Well, that means I need a piece of paper. Okay, so 0.02 rem divided by 100. So it is 0.0002 sievert, or it's 0.2 millisievert. So it's a relatively small dose. So they are estimating that this dose to the childbearing population could impact the mutation rate. Now for us, we have to consider what exams contribute to gonadal dose. Well, barium and enemas, IVUs, abdomen series, lumbar spine series, pelvic, hip series, lower extremities if you're looking at like the femurs. So what this should tell you is just important to protect our patients during those particular exams. In the past, we would have strongly encouraged shielding whenever possible, but today, in today's society, even that is being questioned. But currently, Emory Healthcare policy is still that you should shield, so yes, you should shield whenever possible. But anyway, when we talk about the effects of radiation exposure and mutations on the population as a whole, two things that we look at are the doubling dose or the genetically significant dose. So that finishes up this unit on genetics. In subsequent units, we will be looking at somatic effects, because we talked about somatic effects generically at the beginning of the lecture and then focused on genetic effects. So in upcoming lectures, we will be switching our focus back to somatic effects. So with that, we need to